The embryonic period extends from fourth week to eighth week of intrauterine life. At the end of third week, we have seen that there is formation of a trilaminar germ disc by the process of gastrulation. Now, in the embryonic period, that is from the fourth week, individual germ layer will start differentiating and it will give rise to various structures. So in the embryonic period, we will see the differentiation of all the three germ layer with its derivatives. Now, grossly, the function of ectoderm is protective. So ectoderm is protective in function, endoderm is nutritive in function, and mesoderm mainly forms the connective tissue framework of our body, may it be skeletal framework, musculature, or blood vascular system. They are all formed by mesoderm. Now, the most significant change occurring in the embryonic period is neurulation and appearance of somites. So, neurulation is the formation of neural tube which is induced by the notochord, and this neural tube will give rise to central nervous system, mainly the brain and spinal cord. And secondly, there is appearance of somites. So the intraembryonic mesoderm will differentiate in three parts. Just adjacent to the notochord, we have paraxial mesoderm, then there is intermediate mesoderm, and laterally we have the lateral plate mesoderm. The paraxial mesoderm in post-otic part will get segmented. It will form somatomeres, and these somites or somatomeres will appear at fourth week of intrauterine life. Thus, it divides the embryonic phase in three parts. First part is pre-somite part, which extends from 15th to 20th day. The somites appear at 20th day. So they are seen from 20 to 30 days. So it is known as period of somite. There are 44 pairs of somites, and after 30th day of intrauterine life, it is known as post-somite period. So in this diagram, we will see the differentiation of ectoderm. The ectoderm differentiates into two parts. So this is the surface ectoderm. This entire is the surface ectoderm. Now, if we see in the midline extending from the Hensen's node of primitive streak up to the procaudal plate. So, in this midline, we have a specialized type of ectoderm which is known as neuroectoderm. The neuroectoderm will form neural tube which is induced by the notochord, and this neural tube will give rise to the central nervous system. Thus, we have differentiated ectoderm in two parts. The surface ectoderm, which gives rise to the epidermis and its appendages, and in the midline from the Hensen's node up to the procaudal plate we have the neuroectoderm which gives rise to the central nervous system. Same we can see here, the trilaminar germ disc is there. This is the ectoderm, this one is the intraembryonic mesoderm, and this is endoderm. Now let's focus on the ectoderm. So in the midline just above the definitive notochord, we have the neuroectoderm, and rest of the ectoderm is called surface ectoderm. So here we will see the surface ectoderm. This surface ectoderm is mainly protective in function. It forms epidermis and appendages of skin, may it be hair, nail, sweat gland or sebaceous gland. So in the developing embryo, we have to focus on two areas. So this is the stomatodium. The first area is stomatodium. Stomatodium is the future oral cavity. It is bounded superiorly by four brain vesicle. Inferiorly, we have the pericardial area, and the floor of stomatodium is formed by buccopharyngeal membrane. So if you remember, the procaudal plate forms the buccopharyngeal membrane, and it is the area where only two germ layers are there. We see only ectoderm and endoderm. There is no invasion of intraembryonic mesoderm. So this buccopharyngeal membrane ruptures at fourth week of intrauterine life, and communication is gained between the oral cavity and primitive gut. As soon as this buccopharyngeal membrane ruptures at the fourth week, the amniotic fluid is swallowed by the fetus. It enters into the primitive gut and it nourishes the primitive gut. Okay. Now, similarly, in the caudal end of the developing embryo, we have the ectodermal cloaca. It is also known as proctodium. So, this ectodermal cloaca ruptures at seventh week of intrauterine life. The posterior part of ectodermal cloaca forms the anal membrane. When the anal membrane ruptures, the anal canal gains continuity with the exterior. And anteriorly, we have the urogenital membrane, which on rupturing, the urinary system through urethra will gain access to the exterior, and the vagina, it will again open, the opening will gain access with the exterior. Okay, so here what we have seen, the surface ectoderm, it covers the developing embryo externally. It mainly forms epidermis and the appendages of the skin, and we have seen the two areas, buccopharyngeal membrane and the cloacal membrane. So in this figure, we can see this is the ectoderm. So this one is the surface ectoderm, and in the midline from the Hansen's node of the primitive streak up to the procaudal plate, this area is neuroectoderm. So the neuroectoderm undergoes thickening and it forms the neural plate. So in the midline extending from Hansen's node up to the procaudal plate, the neuroectoderm undergoes thickening to form neural plate. Now, the lateral margins of the neural plate are raised. There are neural folds. There is formation of lateral fold in the neural plate, and a groove or a concavity is formed along the dorsal end. So here we can see a neural groove is formed from the neural plate as a concavity is formed along its dorsal end, and we can see the lateral folds or the lateral margins are raised. Now along the lateral margins we can see specialized type of cells. They are known as neural crest cells. So this is the surface ectoderm, these are the neural crest cells, and this is the neuroectoderm giving rise to the neural groove. Now, along the dorsal end, the, the lateral margins will get fused and it will form the neural tube. So here we can see the lateral margins of the neural groove have fused from cephalic part to the caudal part, and it has now formed the neural tube. There are three important factors responsible for neurulation. What is neurulation? It is the process of formation of neural tube from the neural plate by the process of folding of the neural plate. So this neurulation occurs due to three important factors. So what are these three factors? First is the notochord. The notochord is known as secondary organizer. It induces the formation of neural tube. Then the second important factor is increase in the longitudinal growth of the neural plate. So as longitudinally it increases in length, there is stretching of the neural plate, and due to the stretching, concavity is formed or neural groove is formed. It will further enhance the formation of neural tube. And third important factor is differential growth of specialized areas of the neural tube. So these are the three important factors which are responsible for the formation of neural tube or for the neurulation. So we can see the neural tube is formed by the fusion of the dorsal end of the lateral margins. In this diagram, we can see the formation of neural tube, but at the cephalic part, there is an opening called anterior pore, and at the caudal part, there is an opening called posterior pore. So these pores are present so that the amniotic fluid can enter inside the neural tube and nourish the neural tube. The anterior pore closes at 25th day and the posterior pore closes at 27th day. And then there is formation of dilatation at the cephalic part of the neural tube. So three dilatations are there: forebrain vesicle, midbrain vesicle and hindbrain vesicle. These will give rise to the hindbrain. These will give rise to the brain, and the distal part of the neural tube forms the spinal cord. Thus, the neuroectoderm gives rise to entire central nervous system, peripheral nervous system and autonomic ganglion. Now we will briefly revise the process of neurulation or formation of neural tube. So initially the neuroectoderm gets thickened to form neural plate. Soon a concavity is formed in the neural plate and it will convert the plate into neural groove. The lateral margins are raised, and along the lateral margins you get specialized type of cells called neural crest cells. Once the dorsal end of the neural groove is fused from cephalic part to the caudal part, there is formation of neural tube, and the neural crest cells are now seen in between the surface ectoderm and the neural tube. The neural tube initially shows opening in the cephalic part called anterior pore, and at the caudal part there is posterior neuropore. Anterior neuropore closes at 25th day and posterior neuropore closes at 27th day. Initially these pores are present so that amniotic fluid can nourish the neural tube, and the neural tube now gives rise to the entire central nervous system, peripheral nervous system and autonomic ganglia. And the three factors responsible for the formation of neural tube or for the process of neurulation are: first is notochord. It is the secondary organizer which induces the formation of neural tube. Then longitudinal growth of the neural plate along the embryonic axis stretches the neural plate and makes a concavity, or it helps in formation of a groove. And thirdly, differential growth of specialized type of cells in the neural tube. So these factors are responsible for neurulation. So far we have seen the differentiation of ectoderm into surface ectoderm, neuroectoderm and neural crest cells are the specialized cells present along the lateral margins of neural ectoderm. So now we will see the derivatives of ectoderm. So the surface ectoderm is mainly protective in function. It forms the outer covering of the developing embryo and it gives rise to epidermis and the appendages of skin like hair, nails, sebaceous gland and sweat gland. Also the pituitary gland and mammary gland develops from the surface ectoderm. The sensory epithelium of ear, nose, eyes, may it be corneal epithelium, conjunctiva, lacrimal gland, nasolacrimal duct or the epithelium of lens or retina, they all are derived from surface ectoderm. Again, one point to be remembered is commonly we have seen that all the muscles are derived from the mesoderm. They are mesodermal in origin. Only the exception seen is muscles of the iris and erector pylorem. In the iris we have two muscles, dilator pupillae and sphincter pupillae. So these are derived from surface ectoderm. Also the erector pylorem muscle present in the skin is ectodermal in origin. Then the neuroectoderm gives rise to neural tube, and from the cephalic part of the neural tube, three vesicles are seen: forebrain, midbrain and hindbrain vesicle. They will give rise to the brain, and the caudal or distal elongated part gives rise to the spinal cord. Then we have seen that there are specialized cells known as neural crest cells. They are present along the lateral margin of the neural groove, and once the neural tube is formed, they lie in between the neural tube and surface ectoderm. So these cells will migrate and they'll give rise to various cells. Now, neural crest cells is very commonly asked in Viva, as well as a brief answer question is asked. So we should know the derivatives of neural crest cell. So first derivative is Schwann cell. So these Schwann cells are peripheral neuroglial cells which myelinate the peripheral nerves. Then the adrenal medulla. We know that the adrenal gland has outer cortex and inner medulla, and the medulla has chromaffin cells which secrete epinephrine and norepinephrine. So this adrenal medulla is also derived from neural crest cell. Then the neurons of autonomic ganglion, dorsal root or spinal ganglion, and the sensory ganglia of cranial nerves are derived from neural crest cells. Then the pigment cells of the skin are known as melanocyte. They are present in stratum germinatum and they protect our body from harmful UV radiations. So these are also neural crest cells origin. Then the parafollicular cells of the thyroid gland secrete calcitonin hormone, mainly involved in maintaining the level of calcium in blood. So again, the odontoblast cells of the teeth are derived from the neural crest cells. Now let us see few neural tube defects. So before seeing the neural tube defects, first we will just revise the process of neurulation. So we have seen how the neural tube is formed from neuroectoderm. There is fusion of the lateral margins along the dorsal end, and we have the cranial and caudal neuropores. The cranial or the anterior neuropore should normally close at 25th day of intrauterine life, and the caudal or the posterior neuropore should close by 27th or 28th day of intrauterine life. But if these pores do not close, or if there is no proper fusion of the neuroectoderm and there's no proper formation of neural tube, then it gives rise to neural tube defects. And most commonly the cause of neural tube defect is deficiency of folic acid. So folic acid can be taken by a pregnant woman throughout her pregnancy, and she can start folic acid 3 months prior to the conception to prevent neural tube defects. Now let us see the neural tube defects one by one. So first defect here seen is the craniorachiasis. So in craniorachiasis, there is completely open brain and completely open spinal cord. As the neural tube does not fuse, the fusion of the neuroectoderm does not occur, so there is complete exposure of brain and spinal cord to the exterior. Then the second defect is known as anencephaly. So if the anterior pore of the neural tube does not close, then the cranial vault is absent, the brain is not fully developed, and here the brain is exposed to the exterior because there is absence of calvaria or cranial vault. Then the third defect is the encephalocele. There is herniation of meninges and the brain from the defect. Then anencephaly is in the anencephaly, the occipital skull and the spinal defect is present, and there is extreme retroflexion of the head. So this one is anencephaly. So these are the defects of the brain. Now let us see the defects in the vertebral column. So if there is any defect in the closure of the vertebral column, it is known as spina bifida. The most mild variety is the spina bifida occulta. Occulta means hidden. Here the vertebrae are not completely closed, but there is no herniation of the meninges or the spinal cord. So there is closed asymptomatic neural tube defect in which the vertebrae are not completely closed, but there is no herniation of any structure. Only we can see a small dimple and occasionally a tuft of hair is seen. It is most commonly seen in the lumbosacral region. Spina bifida is common in lumbosacral region. Then the second variant of spina bifida is called spinal dysraphism. Here also more than two vertebral arches are not closed, but here the defect is covered by a lipoma. Okay, so this is known as spinal dyraphism. Now the third degree of spina bifida is known as meningocele. So there is a defect in the vertebral column. The vertebral arches are not closed, and from this defect, either in the spine or the skull, there will be protrusion of meninges, and these meninges are filled with cerebrospinal fluid. So when there is protrusion of meninges through the defect, it is called meningocele. The next variant of spina bifida is called myelomeningocele. So there is a defect in the spine or the vault, and from this defect there will be herniation of the spinal cord, and the spinal cord is covered by membrane or meninges. So this defect is known as the myelomeningocele. Again, there is severe variety known as myelomeningorachiasis. So in this, the spinal cord will herniate out of a defect and it is not covered by the meninges. So here we have seen few neural tube defects ranging from craniorachiasis, in which the entire neural tube does not fuse, so there is complete exposure of brain and spinal cord. Then we have seen anencephaly, in which the anterior port does not close, so the brain is not fully developed, it is exposed due to lack of cranial vault. And then we have seen 5 grades of spina bifida.